Hi, I am Dr. Ayda Başgül Martin and I will talk about blood types today. Please feel free to give feedback by clicking like button or leave a comment below. The major human blood groups discovered by Karl Landsteiner, an Austrian biologist and physician, in 1909. In 1930, he received Nobel Prize in Physiology with his study on blood types. However, I would like to mention the name of the Dr. William Harvey in 1628 discovered the circulation of blood, and Dr. Richard Lower kept the dogs alive by transfusing blood from one dog to another in 1665. Blood groups based on the presence or absence of major antigens on red blood cell membranes. More than 50 antigens have been identified on red blood cell membranes, but the most significant in terms of their potential harm to patients are classified in two groups, ABO and RH blood groups. For ABO group, antigen A and antigen B is the antigens either present or absent. For RH groups, although there is more antigens, but antigen D is the one tested because it has the clinical effect, substantial clinical effect. When you look the ABO type frequencies percentage in the US, you will see the according to gender, Caucasian, African American, American, Indian, Hispanic, and Chinese American, the frequency percentage is changing. But one thing for sure, the most common blood type is type O. Least common or rarest blood type is type AB. That is for all of them. Right? You can see for all of them. Although American Indian has 79% is type O, Caucasian 45%, African American 49% is type O. When you look at type AB, and you see although the percentage is changing, but still they are the rarest type of blood. When we talk about the RH group, 85% of American RH positive. That means they have the antigen on their red blood cell, and the rest 15% has no RH negative, so no antigen D. An antigen is any molecule that triggers an immune response. They are the substances that the body does not recognize as belonging to self, and therefore trigger a defensive response by the leukocytes of the immune system. The antibodies, or in other words, immunoglobulins, are protein-type molecules produced by plasma cells to attack the specific antigen. Plasma cells, a type of B lymphocytes. When you look at the picture, you will see type O has no antigen. You see there is nothing on the surface on the plasma membrane. When you look at type A, you see only the one type of antigen, which is the antigen A, which symbolized or shown with the blue arrows. When you look at type B, there is only antigen B, which is represented by little green circles. When you look to AB, you will see it carries on their plasma membrane antigen A as well as antigen B, both. Because the arms of the Y-shaped antibodies, when they attach randomly to more than one non-self erythrocyte surface, if they have antigen, and they form clumps of erythrocytes. This process called agglutination. The clumps of erythrocytes block small blood vessels throughout the body, depriving the tissues of oxygen and nutrients. As the erythrocyte clumps are degraded in a process called hemolysis, that means erythrocyte membrane is fragmented or disappeared, and hemoglobin inside of the erythrocyte released into the bloodstream. An individual with only antigen A has type A blood. A person with only antigen B has type B blood, and one with both antigen A and antigen B has type AB blood, and one with neither antigen A nor antigen B has type O blood. However, whenever antigen A is absent on the red blood cell, an antibody called anti-A is produced. And whenever antigen B is absent, an antibody called anti-B is produced. Therefore, persons with type A blood have anti-B antibodies in their plasma. Those with type B blood have anti-A antibodies. Those with type AB blood type, neither of these antibodies, and those with type 0 or type O blood have both anti-A and anti-B antibodies. RH was first discovered in a type of primate known as rhesus macaque, which is often used in research due to their blood is similar to the humans. Although dozens of RH antigens have been identified, but only one, designated with D, is clinically important. Those who have the RH-D antigen present on their red blood cells, which is 85% of Americans, and called RH positive. Who doesn't have, we call RH negative. Notice if they have the RH antigen on their cell membrane, red blood cell membrane, there is no antibody designated for them in their plasma. However, if there is RH antigen is absent, it's RH negative, there is antibody, anti-D, on their plasma.